Shall we start?
Yeah, we can start. Am I audible, clear?
Yes, Professor. Good morning, all. Today we are entering our 12th day of our BIIS-12 workshop. On the first session of the day, we have this Professor Vandana Patrawale. She is currently a professor of pharmaceutics at the Institute of Chemical Technology, Mumbai, India. She has around 30 years of teaching and research experience. She has over 200 referred publications with over 9,235 citations with the H index of 49, 22 granted patents, 20 patents in pipeline, and 2 trademark registries. She had published 2 books and 28 book chapters with international publications. Dr. Patrawale has been active in teaching, research, and service throughout her career. She has recently been inducted as an independent director of a device manufacturing company and listed as a top 2% of world scientists in pharmacy. Her areas of research include development of nanocarriers with major emphasis on infectious disease, cancer, and neurodegenerative disorders, medical device development, nanodiagnostics, and nanovaccines. She was awarded Abdul Kalam Technology Innovation National Fellowship 2021, Fellowship of Indian Chemical Society 2020, Mukherjee Oration Award 2020, APTI's Dr. Manjushri Pal Best Pharmaceutical Scientist Award 2019, Shri Amrut Moody Distinguished Researcher Award 2018, OPPI Women Scientist Award 2015, Bill Melinda Gates Grant Award 2015, Best Pharmaceutical Scientist Award 2014, BIIS-12 Award 2013, Minito Nano-tech Award 2013, APTI Best Teacher Award 2012, Fellowship of Maharashtra Academy of Sciences 2011, and KH Garda Distinguished Researcher Award 2009. She is Vice President CRS Indian Chapter, Editorial Board Member DTTR, Editor CRS IC, and APTI Women Forum newsletters. She is actively collaborating with researchers as well as industries within country and abroad, and has completed Indo-Swiss, Indo-Japan, Indo-UK projects. She is executing all major grants from Indian government focusing on product development. She has transferred about 20 technologies to the industry, including drug-eluting stents being marketed in more than 65 countries. So with this short introduction, I invite Professor Vandana to deliver the lecture. The floor is yours.
Thank you. Thank you for the kind words. And right away, I think I'll start with my presentation. And if you could confirm if my slides are visible.
Yes, Professor.
Okay. So great friends. Really nice to meet youngsters on this platform. I've been working as was introduced on nanotech based drug delivery systems. So today we look at what are recent developments and prospects. What I intend doing, of course, is give you some case studies which we have done at ICT. And ICT, that stands for Institute of Chemical Technology, which is located at Matunga, Mumbai, but now has 2 more campuses, one in Jalna, Aurangabad, another one in Bhubaneswar, Odisha. So here our mandate is that we work on technologies which can be transferred to industry. So that means you have to keep it very simple, cost effective, and look at what industrial houses have so that the transition can be smooth. Of course, we are working on basic sciences also. So we do use this approach, which is integrated approach in science and technology for sustainable growth. Now coming to what is nano, I'm sure you all know. When we say nano, typical nano systems would be only 1 to 100 nanometer. And 1 nanometer is 10⁻⁹ meters. So 1 billionth of a meter is a nano. So that says about the scale at which we are converting our active pharmaceutical ingredients or for that matter, if you are looking at any of the systems, be it lipidic, polymeric, inorganic, to this scale. So in recent years, ability to manipulate matter at this scale really has increased a lot because of the obvious advantages, and you'll see in coming slides how. When we talk about nanotherapeutics, 1995 saw many different type of nanotechnologies being approved. So obviously, people did start with polymeric particles, but when you're making polymeric particles, the residues of monomers and solvents can be a major issue. And therefore, people shifted to lipidic systems. And in lipidic systems, because lipids, daily you are eating, they are part of your body, they are biocompatible, biodegradable. There are obvious many advantages. So 1995 saw introduction of liposomes, which are nothing but vesicles. They are bilayer vesicles in which the drug can be incorporated. Both hydrophilic drugs as well as hydrophobic drugs can be incorporated, because you have inner aqueous coat and then you do have a hydrophobic coat, and could be vice versa also. Amphotericin picked up, that in recent times you would have heard, in pandemic time, amphotericin as a drug, which is used for many fungal infections, used for many bacterial infections too. So lipidic discs were formulated. People went on and worked on many of the inorganic nanoparticles. So be it iron oxide, be it gold nanoparticles, be it platinum now, silver nanoparticles, because of its obvious benefits as antibacterial, and started loading them with drugs. And now this field has again grown, because what has come into picture is nanotheranostics. Now, what are theranostics and nanotheranostics? Basically, they are looking at therapy as well as diagnosis. So giving dual advantages, and therefore they are very, very useful in case of, say, if you need to track where the cancer is and over a period of time, whether the medicine is being delivered at the site or not. So nanotherapeutics is now picking up. Then people moved on to simpler systems like polymeric micelles. Micelles, as you know, are made up of something which will have a hydrophobic chain and a hydrophilic polar end. So they can actually self-assemble to give many, many advantages. So around 2000 to 2005, many people started working on these kind of systems, especially if you look at certain hormones which were encapsulated to treat menopausal symptoms. Then there are moieties which cannot be, in fact, processed by any of these techniques for simple reason, because the drugs which are of very high melting point, having very high log P value, which actually is not amenable to making complexes also. So there nothing remains but to process them as such and make nanocrystals. So there are many marketed products in this category also. They do not use any of the lipid, they do not use any of the polymers except for stabilization, small amounts. But otherwise, majorly, this is only drugs which are processed to give nanocrystals. And then there were virus-like particles. Now the era is that we are actually focusing on cell-based therapies. And obviously, again, we have seen re-entry of lipidic systems. The mRNA vaccine, which was talked about by many during COVID period, is lipid-based. So it is nano-based, and in that nano, lipid is being used actually as a formulation ingredient. Looking at the commercial aspects, again, global market is really growing by 14.5%, and you see the projected market in 2027 is $475 billion. So that itself says that this is growing at a very, very rapid rate. So initially people were working on something which is called as passive targeting. When we talk about passive targeting, you are giving, say, intravenous injection. It is a parenteral administration, and the drug, by its virtue of size, will reach the tumor. And why does it reach the tumor? Because when you're talking about tumor, the vasculature has leaky vessels. These angiogenic tumor vessels are leaky and they are permeable. So nanoparticles can travel to the tumor site and get lodged. But now we are talking about something known as active target. And when we say active targeting, typically you do have ligands which are attached to these nanoparticles by making use of a ligand. And when you are giving intravenous injection, they get localized only in tumor. That means they are not going anywhere else in the body, but because these ligands have specificity, because of the receptors which are overexpressed, say in cancer, or for that matter, it could be anything, could be any of the CNS diseases, could be any of the serious diseases, so on and so forth. You need to decide which ligand one needs to attach to these drug-loaded nanoparticles so that it goes in a site-specific manner to that particular place only. The net result is you do not get any of the side effects which would have been seen with passive delivery. So now moving on to some of the case studies which we have done. So first case study is on nanostructured lipid carriers, which we have formulated first time in the world for cerebral malaria. Now, why did we select cerebral malaria? Now, here what happens is if P. falciparum affects any of the human beings, you know, it is transmitted, malaria is transmitted by mosquito as a vector. So if that happens, generally the patient becomes comatose if immediate treatment is not given. And marketed formulations which are available are not very stable formulations. So first we looked at the statistics. So we know that yes, India does get affected, about 2% the malaria deaths are in India. Major, of course, is the African countries, but 2% of Indian population is a huge population. So there is an unmet need still. And then we looked at what can we go ahead with. We try to understand how the normal RBCs look like and what happens when RBCs get infected. Having understood the pathophysiology and knowing that there are certain changes which are happening on the surface of RBCs, we thought, can we design something which can effectively get delivered very quickly to brain? Because it is in the brain that these vessels become very hardened, as a result of which, you know, the blood cannot flow through it. And that is what is leading to coma-like symptoms. So then we looked at the WHO guidelines. It's very important. Those of you who are thinking of working in this particular area should be really updated about what WHO has to say. If you see the guidelines very clearly says that you should not be using monotherapy. And what is there in the market today is only monotherapy. That means single drug. Why monotherapy should not be used? Because it produces resistance. So over a period of time, it will be useless. So it's always good to use it in combination. And in this category, we have something on the top, which is artemether and lumefantrine combination. Now, this combination is available as oral dosage form, but because of inherent properties of artemether and lumefantrine, no parenteral or injectable dosage form was available. Oral, you know, it has to be taken by mouth, reach stomach, reach intestine, get absorbed, and then it will give effect. And here we are talking about cerebral malaria, where hardly there's any time for patients to survive. So this is a recommended combination therapy. And when you look at what is written at WHO site is mortality from cerebral malaria is 100%. So this gives us more reasons to work on something which can act quickly. So we formulated NLCs. For those of you who do not know what are NLCs, as I mentioned, they are nanostructured lipid carriers. As we said, we use very simple technology. So we just take oil, surfactant, and co-surfactant mixture. They have to be chemistry-wise similar. Put our drug in it and dilute it with water, and what you have is NLC. So those of you who think that it's very difficult to make nanosystems, please do not be under that impression. We hardly have to use one in a large scale, only a stirred tank, nothing else. No high pressure homogenizers, no microfluidizers, no sonication, nothing at all to get nano. So we prepared those systems and looked at whether they are taken up by RBCs or not, and that also by infected RBCs. So obviously, these experiments are done in animals, specifically mice. Mice cannot be injected by P. vivax or P. falciparum, but when they are injected by P. burgii, that's Plasmodium burgii, it gives you malaria-like symptoms. So that's a very, very well-established model. So you can see that when we are giving our NLCs, that's nanostructured lipid carriers, there is no parasitemia. That means infected RBCs, the P. burgii gets killed, as compared to when you see that no treatment is given. And when we do that with the combination, in fact, absolutely sustained release manner, the drugs are getting released. There is initial burst, which is required also to take control. And then over a period of time, the drug gets released, which is also a required phenomena. So that condition does not come back. In terms of science, they are known as recrudescence. So we don't want recrudescence. The particles, when they are characterized, you can see are about 60 to 100 nanometer in size. So having done this experiment with combination, we wanted to see how low can we go. If therapeutic dose is say X, can we inject 1/5 of the dose, 1/20th of the dose, 1/50th of the therapeutic dose, because we are converting them to nano. So is there a chance that we reduce the dose? Is there a chance that we reduce the frequency of administration? Answer to both these questions is yes. Because as can be seen in this slide, even at 1/20th of the therapeutic dose, we can see that there is no parasitemia, even 21 days post-infection. Whereas what you can see here, of course, is what happens with the marketed formulation. So till the time you're administering marketed formulation, parasites are under control. You remove the therapy, and again, parasites grow, which doesn't happen in our case because our system is very, very specific. Then we did it in malaria-like model. That means you allow the symptoms to appear. Don't give therapy right from the start. So about 10 to 20% parasitemia should be reached before you can initiate the therapy. Again, we saw that our system, even at 1/10th dose, has shown complete parasite clearance as compared to all other groups. It was a real good point to move ahead, and it is at that stage that we have taken a patent for this particular preparation. In fact, that patent is a very, very wide patent. It is nano drug delivery based on combination therapy for treating parasite infections. It's a granted patent, and the technology actually has been transferred to industry. Now was the question whether it is acted actually in malaria cases. What would happen if we give it to cerebral malaria? And again, you can see that as soon as a therapy is given, parasites are under control. And in this case, this study has to be done actually in C57 mice. All these studies are done in Swiss mice, but in this case, it has to be C57, which is a special kind of mice, black mice. Study has to be done. And yes, You can see what happens with the marketed parasites grow, but with our formulation, they are under control and completely cured. In fact, our vet scientists, that's veterinary doctors, said that they are so active that can be used for another study. Of course, we don't do that for ethical reasons. And here it would be very, very clear. Now, the formulated system, before I start this video, you should know that we have labeled it with rhodamine. The NLCs are labeled with rhodamine. Wherever you see granular mass in the RBCs, they are the ones which are infected. But you will see that when I start the video, these NLCs are taken up specifically by where this infected RBCs are. The plain ones, RBCs, which are normal RBCs, I suppose you know as biology students that they do not have nucleus. The nucleus will be only there when you have infected RBCs. So when I start this video, it will be very clear that NLCs is a live video. NLCs are taken up only by infected RBCs. So very, very selective phenomena. And the selective phenomena is not only due to nanosize, but it is also due to the chemical composition of our NLCs. And we proved that by variety of a test, and which is actually listed in this paper, which you can see at the bottom of the slide. It is in International Journal of Pharmaceutics, as well as it's there in biomaterials where we have indicated. Now you can see what happens in the marketed group. This is after treatment. I told you that coma-like symptoms. So in the C57 mice, they are sort of paralyzed. So after treatment also, you can observe their gait. They are hardly moving. So they are not active mice at all. And that happens at the therapeutic dose. Whereas in our case, even at 1/20th of the dose, these mice are as active as normal after treatment. That means the symptoms are totally reversed. So this, of course, has given us enough confidence to move ahead to clinic, and definitely even 55 days post-infection. What is very important in such cases is not only that they are efficacious, there should not be any casualty seen, no mortality seen. So the mice in our group, they survived up to 55 days post-infection. In fact, that was a period for which we observed them, which is the normal maximum period for which any animal study would go on with this kind of mice. Coming to the next study, and that study is on breast cancer. Our lab works majorly in 3 areas. One is infectious diseases, another is cancer, and third is neurodegenerative diseases. Now again, look at statistics. Breast cancer affects all kind of women, meaning women in any of the age groups. It could be as early as 20, it could be as late as 80. All are at risk of cancer. And in fact, if you see these statistics, no metro city is spared. And incidence is just growing. By 2020, about 1.24 lakh people, women, are supposed to get breast cancer. In fact, it's already surpassed this figure. It's more than double than what was projected. Reasons are many, but look at the statistics. 1 among 8 women are at risk of breast cancer. And annually about 1 million women are diagnosed. 5 lakh about die from this disease. So why is this happening? Try and understand that major reason is metastasis. Tumor may be there in breast, but it gets actually conveyed to various other organs. And that happens because of the effect known as angiogenesis. What is lymph angiogenesis? Many of the lymph vessels grow. There are newer lymphatic vessels. They grow when women gets affected by breast cancer. Net result is The sentinel lymph node, which is sentinel lymph node is a lymph node which is next to the breast, that becomes the housekeeper for all these cancerous cells. And from this sentinel lymph node, it actually spreads to various organs. It disseminates these mets, as they are known, through systemic circulation to lungs, to liver, to bone, to brain, because of which there is decreased patient survival. So our hypothesis was, can we make a simple system? And since we are focusing on lipidic systems, which gets localized in this sentinel lymph node. So if it kills everything here, there is no chance of reaching all other organs. So with that hypothesis, we started and we made what is known as imperfect NLCs. I've already told you what are NLCs: oil, surfactant, co-surfactant, diluted in water. Imperfect ones are those which contain more of liquid lipids. As a result of it, some of the drug is concentrated in liquid lipids, most of the drug is in solid lipids. Why do we want such a thing? Because whatever drug is there, the liquid lipid will come out first. So it will take care of initial burst and rest of it will slowly come from the solid lipid. So we have specialized in making these NLCs. And when these NLCs will be taken by oral route, what is expected is they will go to the intestine, and from the intestine, they'll be taken up by endoplasmic reticulum, go to the lymph first, and then they will go to the blood circulation. And once this happens, that they are going to the lymph first, that means they'll go to sentinel lymph node first. As a result of which, it will not give the normal toxicity which is associated with these drugs. So such systems are known to enhance not only bioavailability, they are supposed to reduce any kind of side effects. So the drug which we selected for this is the first line anti-estrogen agent, which is being used for breast cancer. That's tamoxifen citrate. But currently also, it gives all these, which you can see on the right side, all these symptoms which are toxicity related. In addition, it undergoes extensive first pass metabolism, which necessitates higher dose to be given to be effective. So we thought by using the nano system, will we be able to reduce the dose and therefore reduce the toxicity as well as reduce all the kind of side effects which are seen. So we formulated the system, as I told you, by selecting right kind of oil, surfactant, co-surfactants. How do you select those? You have to select the oil and lipid in which it has maximum solubility. The drug has maximum solubility. You have to select co-surfactant, surfactant in which it has minimum of solubility, but it gives you NLCs. Now, why it should have minimum solubility in surfactant, co-surfactant? Because if it has maximum solubility in it, over a period of time, your drug will leach out in the system. We want the robust system and should be a stable system. So do the studies to identify in which solid lipid and liquid lipid drug has highest solubility and which surfactant, co-surfactant has lowest solubility, yet the system is stabilized. So we did cell line studies, and you know, for breast cancer, it's MCF7. And what you can see is that we also did studies in ZR75-1. We always tend to do one more model everywhere just to be doubly sure. Now, what is the difference between MCF7 and ZR75? This is mucus bearing cell line. So in case, you know, there is some hindrance offered, yet whether our system is giving us the effect or not would be very clear. And you can see that cancer cells definitely are getting killed by our NLCs. You may wonder and say that Tmex, that's your tamoxifen alone group, is better. But in fact, Tmex, as you know, is very low solubility. What we have done is solubilize it in DMSO. So what we can say is molecular solution of TMX is behaving just the way our system is behaving, which is a great sign. Because in actuality, you will not have the system. You cannot use DMSO internally. So our nano system is behaving like atomic system and working really, really well. So we did cell uptake experiments, and what you can see here is confocal images. It was very clearly seen. What have we done? Now we have labeled our NLCs, in this case, fluorescent label. So what you see as blue is DAPI loaded nucleus. So DAPI is a dye which can stain nucleus. And NLCs are majorly localized. You can see in cytoplasmic region, perinuclear region, and even the nuclear spaces. And again, this was confirmed by flow cytometry. In flow cytometry, one cell goes at a time, and you can see there has to be a shift in this graph. So if cell population initially is in the control like this, you can see that it is shifted. That means all the cells have died. So it is shifted. So that gives us an idea that yes, there is a nuclear co-localization and cell death happens with this. Having done that, we wanted to proceed to in vivo experiments. So we did pharmacokinetic studies. And here you can see suspension, which is the marketed product versus our product. So our product has given very high area under the curve, which indicates that it is more bioavailable. But what you can also see is what students saw and reported to me, there's a lag. There is a lag as compared to the marketed system. I said, good. Why? Because this is indicating that, yes, they are going first in lymph and then coming in blood, because these studies are plasma concentration studies. So definitely looking at this, we have seen that T half is prolonged. That means as compared to the original system, which will be there for 5 hours in the body, this will be for more than 25 hours. Why more than? Because this is T half. Half of the amount by 25 hours is detected. That means it will still prolong. So it is again a good sign. And more than this, what was important was this study, which is mesenteric lymph node uptake study. We wanted to see, okay, blood it is reaching, but whether it is reaching in lymph node. And this is what astonished us. Again, it is because of this that this has gone to clinic. What we do at ICT is we generate proof of concept, identify industrial partner. We do all kind of toxicity studies, stability studies, these kind of in vivo pharmacodynamics, kinetic studies, and pass it on to industrial partner. So what you can see is with the marketed, nothing in the lymph node. Whereas with our system, at least till 4 to 6 hours, you can see about 160 nanogram per milligram, which is a very high amount in sentinel lymph node. And it has reasons for us to believe that would be a real good system, because we have concentration, the sentinel lymph node, which is much, much higher. That can be seen here also. So when we actually did the studies by generating tumor in animals, you can see that even half the dose of NLC is much more effective than marketed. Now, marketed, what are the issues? Is it not effective anti-cancer agent? Yes, it decreases the tumor burden. But what happens, it has many, many side effects. So we wanted to see that, yes, it is definitely significantly reduce the tumor volume, but whether our system is also safe. So first thing is survival plot. You can see in purple that 100% of mice in our group have survived. At 3 mg per kg, they have survived. As compared to that, marketed, you can see this is showing that mice who died. So 12.5% of mice survived in case of marketed formulation versus 80% surviving in our case. Very good sign. Go to organ toxicity, do histopath studies, and again you can see that TMX NLC, that's tamoxifen NLC, is very, very safe. In case of marketed formulation as well as a plain suspension, what was seen was all kind of congestion, whereas in our case, no abnormality was seen. So subtherapeutic dose of tamoxifen in NLC was curate. Okay, it cured all the mice. In fact, none of them died, no side effects seen. Then we went on to see whether this similar concept of NLC can be used for intracellular availability. Now, when we are talking about intracellular, nowadays there is a trend to give nucleic acid delivery also using these lipid carriers. That's how your mRNA vaccine also would work, right? So here we started with one small moiety and one large moiety. Idea was whether our system is able to give transfection, whether it is able to prevent lysosomal degradation so that it can have endosomal escape and therefore it can enhance the transfection ability. So in this case, we have synthesized a cationic lipid. The same NLC platform, we designed a cationic lipid. Again, it is patented, and since I have given it to industry, I cannot reveal, but I will tell you the concept. What was in our mind is, if you have a cationic lipid, it will interact well with any of the membranes in your body. Why? Because all the mucosal lining in your body are negatively charged. And opposite charges attract. So cationic lipid, it will interact with anionic lipid in the body. And then there will be a flip-flop phenomena. What does that mean? If our cationic lipid come across a different kind of pH, it will give a total reversal of the structure. That means there'll be reorientation of this lipid so that the moiety can escape. So initially there is a stable cone complex formed, and then there is you can see there's a total reverted structure which can lead to destabilization of endosome, and that happens, the drug will escape, and drug escapes, that means it's near nucleus or within the nucleus. And this is true for any cargo. It could be drug, it could be siRNA, it could be any of your protein, it could be even mRNA. So this is how our cationic lipid looks like. It has a sort of rheological property where it decreases with increasing shear rate. That means it is pseudoplastic. It's also thixotropic. So we formulated now non-cationic Smeds and cationic Smeds. You may wonder what are Smeds. So Smeds are self-micro-emulsifying drug delivery system. Again, they are nano range. In earlier case, we had oil surfactant, co-surfactant diluted in water to give you NLCs, right? In this case, they are not diluted, they are used as such. So it's just a combination, just a mixing of oil surfactant and co-surfactant. In this case, we have added cationic lipid. So obviously, when you add cationic lipid, you expect that it will have a positive charge, which was actually confirmed by using a zeta meter. So zeta potential of this system is plus 32 millivolt, very stable system. And for those of you who are wondering, because I've not given you what cationic lipid is, I can just say that in this case, we have a range of lipids synthesized. In fact, even many of the vaccine companies are looking at interacting with us and taking this. It has cationic guanidine group. So we call this as a heterolipid. It is monoguanidino heterolipid. So in this case, we have used etoposide as a moiety. So we looked at melanoma cell lines, and again, you can see that etoposide is really very effective when Smeds are given, and very, very effective when you have a cationic Smeds. So ECS stands for cationic and NCS stands for non-cationic. So non-cationic is also better than marketed, but cationic is much, much better than marketed preparation. And by, again, morphological alteration, we could see that is very, very effective. If this is control, you can see that all the cells, almost all the cells are killed by ECS. This is one very interesting assay which we perform, which is generally done when people are taking radiation. When any cells are irradiated, they will be killed. But in case of cancer, again, these cells grow. So we thought, can we do this in vitro test? We can treat it with plain etoposide, that is a drug, non-cationic, and with ECL. And to our surprise, the ones which were treated with cationic Smeds did not allow any cancer cell to grow again. So this, what you can see in slide, is after 14 days. So this plate is very, very clear, even after 14 days. So, which is also represented here, which is a very, very good sign that the system is effective. Then we wanted to see how are they taken up? How are they taken up intracellularly? So for that, you do have different conditions. You can cool the cells, you can give chlordiazoxide treatment, can use colchicine, you can use sodium azide, genistin, and each one is different. So if you are, say, example, treating the cells at 4 degrees Celsius and by sodium azide, you can make out whether it is taken by energy-dependent pathway or not. This will give you an idea about clathrin-mediated pathway. Colchicine will give you an idea about cytoskeletal reorganization, so on and so forth. So wherever you can see your NLCs or Smeds, in this case, cationic Smeds, getting localized, that means that particular pathway is not very effective. So in this case, it was cytoskeletal reorganization and energy-dependent pathway by which the drug was taken up from Smeds. Then we did in vivo anti-tumor studies. And again, in this case, you can see that our system was much more effective, and that also at the subtherapeutic doses. Most of the cases, we have reduced the dose, reduced the frequency also. In some cases, you will be surprised that we've gone to 1/50th of the therapeutic dose. So by that means itself, you are reducing the dose, you're reducing the side effect. And then if you have active targeting, you are reducing even frequency of administration. And that's what we have to look at, as far as patients are concerned. Survival plot again indicated 100% survival in our cationic Smeds versus 25% of mice surviving in marketed formulation. Toxicity studies, when we did, if you give the marketed formulation, you can see all kind of abnormalities in RBCs. But with our system, intratumoral delivery, no abnormality seen. So we attributed this, of course, to its high transfection ability and nano size driven increase intracellular bioavailability. So it's not going in any other areas, and because of which it is not giving you the side effects. Same thing, we did it with plasmid. And if we can do it with plasmid, that means we can do it with any kind of nucleic acid delivery system. So we could actually overcome all the barriers which such systems will have. Which are the barriers which it will have? There are DNAs, there are nucleases, there's lysosomal degradation. All of this has to be prevented if you have a robust system. So we generated Monobinoplex. Why I say Monobinoplex? Because my lipid is guanidino, right? So I've taken a solid lipid, surfactant, and my cationic lipid. And this is what you get, cationic solid lipid nanoparticles. Incubated with any of the plasmids. In this case, we've taken P53 because green fluorescent protein, so easily you can observe. And you could see that it gets intercalated because of the charge-based phenomenon. And you have to make sure, you can see here that if you have naked DNA, in presence of DNAs, no band would be seen because it totally destroys it. But when I have my SLNs, in presence of DNAs also, you can see the band. That says that moiety is within the SLN. It is not out. And therefore, DNAs is not able to degrade it. So, of course, you need to do some studies to see what would be the ratio of SLN which will be used in comparison to your cationic with respect to your nucleic acid, which you want to encapsulate. So these studies indicate that Our Monobinoplex is similar to lipofectamine. Lipofectamine is actually a gold standard, but this cannot be given internally. It has toxicity issues. So we have now a tiny SLNs, in fact, which can be administered internally. So this opens up a menu for us. We can use it as vaccine adjuvants also. In this case, of course, we have encapsulated P53 and looked at the transfection assay. You can see this is how lipofectamine gives transfection, and Monobinoplex is similar to lipofectamine. So this type of system again can be used for nucleic acid delivery. Now, what other people are doing now? A lot of work we are also doing, but I'm not presenting the data here. You can modify nanoparticles. You can use nanoparticles for delivering antibodies. Nucleic acid delivery, already I've shown you some example. You can go on modifying the antibodies. You can have Fe bodies. You can have smaller antibodies and then put them as ligands on your nanoparticles. You can deliver cytokines also using nanoparticles. In fact, people are doing pegylation. That means they're treating it with polyethylene glycol. This makes it long circulating. So pegylated cytokines are also available. And from there on, we are moving to, as I told you, now the trend is personalized medicine. That means CAR T therapy. And you'll see probably in the next slide. But before I go to that CAR T, this field is growing too. So needles, parenteral, many people are scared. Now there are microneedles. So microneedles, they traverse only epidermis. So when you apply them on skin, it would be like you applied Velcro, just like your Band-Aid with little Velcro. So you can make different type of microneedles. They can be of silicone, metal, polymer, ceramic, silica glass. They can be dissolvable, and that's what we are working on. That means you apply, microneedle dissolves, leaves a cargo, and cargo forms a depot if it is required, and then it slowly goes into the blood circulation. If faster onset is desired, it doesn't form a depot. So your system has to be such that immediately it penetrates. So different kind of nano delivery systems people have used along with these microneedles. There are various techniques of synthesizing these microneedles. Nowadays, you get ready molds, and all you have to do is, these kind of molds, you need to fill it with whatever polymer drug is, and then get the kind of needle you are interested in. So a lot of work is happening in this area also. You do have smart stimuli responsive media nowadays, microneedles. Why I say stimuli responsive? You can apply exogenous stimuli like from outside, like ultrasound, electric current, so to release the cargo, or it could be endogenous stimuli. For example, in this case, if you have microneedles of insulin, they can sense what is the blood glucose concentration, and then they can deliver whatever amount is required. This is a big, big field. Many people are working on this particular endogenous stimuli. Not only through skin, you can give it. Ocular micro needle based delivery is also possible. It's very difficult to reach posterior segment, and people take injection. Those of you who are aware, there are certain disease conditions which affect the posterior segment, back of the eye. You don't have any other option but to give injections in the eye. So in those cases, such simple microneedles, as you can see here, very tiny ones, can be delivered, and they will actually deliver the cargo to the back of the eye. And this is what I was talking about earlier. CAR T cell based therapy, there are already products in the market, especially for leukemia, blood cancer. So what is done here is you're removing T cells from patient's body, growing them, inserting the gene which is for CAR standing for chimeric antigen receptors. So you need to identify that receptor and accordingly insert the gene, grow it, again infuse the same thing into patient. So now patient's own body cells are engineered and then they will tackle any kind of cancerous cells within his or her body. Latest in this is CRISPR. It's similar, but much easier. And what people have been doing is customizing this particular technology. It gives you rapid gene editing, very easy to scale up, cheaper. So we are moving towards personalized medicine. The procedure is actually almost the same as for CAR. In this case, you are making CRISPR edited T cells in lab. And that would be injected. They are also being used for cancer mainly. Some nano devices are there. For example, this particular device, if you've heard of Japanese technique called Origami, so this will, on certain stimuli, will roll up. You can fill it in capsules, or you can give it by injectable route. And when it senses environment changes, for example, in this case, pH, it will again unlock. So when it unlocks, it will release all the cargo which is onto this device. Okay, again, this particular thing is being used for many of the infections, many of the tumors. Extracellular vesicles are becoming important. See, people thought that we'll make, earlier people were using virus-like particles or using viruses which are inactivated. Now they are using all kinds of cells. Now these cells could be monocytes, could be RBCs, could be anything. And you can engineer them. You can engineer them by, say, coating with PEG. You can have glycan groups. You can have peptides. You can have other ligands. You can attach antibody, just the way I told you earlier slide, that they can be modulated as per the need. And they can encapsulate any kind of moiety. It could be small moiety, it could be large moiety. It could be chemotherapy drug, could be proteins. So a lot of work is going on in MIT and of course in India also in many places, including in our group on extracellular vesicles. Of course, we have not reached the bedside yet, as with other cases, but here, this is really, really promising. So nano constructs definitely can overcome all the drawbacks of conventional cancer therapy. You can use certain biomimetics, just like in the last slide I told you, any kind of cells. You can use these for bacterial sensing. You can use externally stimuli. Nowadays in vaccines, a lot of work is being done on artificial intelligence, meaning which kind of mRNA will interact with which kind of lipid or polymer can be done by computer aided. And then you can design them and inject them. Beat anything. Generally, we have seen that academic institutions reach only TLR3. Technology readiness level is TLR. Or what we want is take it all the way ahead. So what is expected of you all is you have established proof of concept, do early stage validation, late stage validation, see how can it be commercialized and then enter into commercialization. So it's a long way, but you need patience and perseverance. And I'm sure if you have idea, one idea can change entire thing. For example, this cardiovascular stents, which we have made for a company called Surgeon Medical Technologies, is now in more than 65 countries. What was our idea here? Idea was that stents, which are used as a replacement for bypass surgery, when vessels are getting clogged, the vessels are forming plaques, because of it, the blood cannot run in the arteries. So to open up through the thigh, you are inserting something like this, you have catheter on which there is a balloon, on which there is a device, which is spring-like device, which is known as stent. So when you inflate the balloon, after it reaches a place where there is plaque, inflate the balloon, this will get expanded. So your spring-like structure opens up. And after that, deflate the balloon and remove it, the stent will remain in place. So what have we done? Our group has loaded this with microspheres and nanospheres. So this particular stent remains in place. The nanoparticles which get released from this, they go into the intima and media of the blood vessels and get localized there. And then they give the effect. So there is a video, but I will skip that in interest of time. What I will show you is what we got. So we have loaded drugs like sirolimus, and you can see that percentage incidence of all the kind of problems is definitely reduced. So we are getting with sirolimus loaded systems very, very good protection. And this has been shown by optical coherence tomography also. So here you will be surprised that the study was done versus Abbott stent. All the studies are done in Europe. There's a battery of tests which have to be done. I'm not showing that today. Only one thing, that is tomography, which indicates that our stent is equivalent. Rather, in our 5-year results which we have got, it's better than Zion stent. Very soon we'll be entering US market also. And that is the kind of thing which you can do. What was our idea? Our idea was that we'll use biodegradable polymers. None in the world was using biodegradable polymers. Everyone was using bioresorbable, rather bioerodible polymers. And we thought if bioerodible polymer is used, it will give out small moieties of polymer over a period of time. They themselves can create issues. So why not have something which slowly dissolves? There are no issues. End products are like water and urea. And with that, we designed the coatings, and now world over, people have started using our technology. So now we are extending this technology even to balloons. There are many conditions which do not require stenting. So with simple balloon, if you can coat the balloon surface, this can be done. So now results very soon would be out. Already studies have to be done in pigs. You can't do it in small animals. These studies are in pigs, so we do it in Europe, Netherlands specifically. And I'm sure even they'll be going to market. So obviously, from bench, 1997 we started, 2005 we introduced first stent. And from 2005 till date, there are 8 stents which have come in market. More than 3 lakh stents have been implanted. So we are learning from our own experiences. We are making it better and better. When we started, it was a 4-layered stent. Now it is 2-layer stent. And we hope that with biotechnological advances which are taking place, we may be able to replace this small molecules which are chemically synthesized with even biological moieties to permanently tackle. Many cases, you'll be surprised, in one body, there are 6 to 7 stents which are implanted. One thing which we have achieved by doing this is we have reduced the cost. So if in market, Abbott was being sold at 3 lakh per stent, we have reduced it 5 times now, in fact, even 10 times, because it's come under DPCO. These are some of the stents which are there already in the market with our expertise. From domestic business, we are having about 46% of revenue, but from international, currently it is 54% of revenue. So can you imagine that something which is done by mere idea? It started as a mere idea, very important story, because these people had lasers with which they were cutting diamonds. So they came to us to explore what more can be done. And one idea was whether stents can be done. And I said, why not? We did not know anything about it, but we read about it, and I directed them, gave them a paper model as to how this could be done. So they employed mechanical engineers, they employed all kind of engineers so that they could make the base metal stent with different designs, with lock-in place. Now we have lock-in place, and we have the best design. In fact, world over, our stent strut thickness is the lowest, And that is what is required if you want minimum of, again, side effects, minimum of problems. And our idea, how to coat it, which particular polymers to be used, and what proportion drug to polymer should be used, really has worked very well. So we are hopeful that the same technology, when it comes on balloon, it would be really, really very great. Another thing which we are working on is on unmet challenges. Currently, if people have diabetes, or ulcers, diabetic ulcers, very difficult. You have to have stent, which is of a very, very small strut thickness. So now we are working in this area so that we'll be able to solve this kind of problems also. And the release of the drug can be for 1 to 2 years rather than conventional 1 month. So a lot of challenges, but I'm sure all of you will be able to take some of these challenges and take it ahead. There's no approved drug-eluting stent for peripheral use. This itself says it all. So if something can be done in this direction, it would be great. So I have a Prime Minister fellow now in the lab working on this particular technology. So if it was not for these collaborators and supporters, I'm sure we would not have done the kind of work which we are doing. Our lab is equipped with each and everything which is required to make any kind of nanosystems and characterize them also. And without this, my team, I would be definitely not be talking to you. Very, very dedicated students. So everything what you have seen, I dedicate to these students who have worked really very hard to come to this level. So with that, I would like to end and take up any questions which you might have. Sorry for 2 minutes extra.
Thank you so much, Professor. The session is open for discussion. Any questions?
Feel free to ask questions. Otherwise, you can write to me by mail also, and I will be able to answer. Maybe organizers can share the mail ID.
Sure, Professor. I will share.
Okay.
Questions?
So have the next session
starting. Yeah, starting in 10 minutes.
That is the idea, Anjali, that give you something new so that you can think in that direction. Thank you. The compliment. I think you can see my mail ID in the chat box. Parthipan. Okay, no questions. So I would just say all the best to all of you for your future.
Regarding the placebos.
Archit said regarding the placebo, yeah, very important question. In fact, that has opened up a very new field in our case, in case of malaria. Thanks, Archit, for pointing out. In case of malaria, the system which we have, only the blank, that is placebo without the drug, is performing better than the marketing. And that opens up eyes for everyone. And that's because these nanoparticles have ability to be taken up by tubovesicular system, which the parasite has for taking its food. So now lipid is a food. So it gets confused and takes our nanoparticles in. So when it takes nanoparticles inside it within the RBC. Now our nanoparticles, they have a mechanism by which they generate a lot of reactive oxygen species inside. The mitochondrial calcium comes onto the surface. It just reverses, because of which the parasite dies. So we have looked in detail, and if you read that biomaterial paper, it would be very clear to you that it's a chemical composition which is leading to this particular effect, and because of which the parasites die. So what we are trying to say is that you now have a platform, and in this platform, any kind of antimalarials can be added. Or for that example, anything which has to target RBCs can be encapsulated so that it would work even better than any of the drug combinations. So thanks for asking that. Placebos do work, specifically if they are in nano range, and specifically if they are being thoughtfully designed.
Any other questions? If no more questions, then I would like to thank Professor for joining us today.
My pleasure.
Thank you so much, Professor, for sharing your time and knowledge with us.
Always a pleasure. I'm always available. For Srishti, for BIIS, for DPT.
Thank you.
For my nation. Yeah, thanks.